Hello, everybody. In this lecture, we're going to talk about how to grow microbes and specifically growth media. And so by the end of this module, you should be able to describe the three formats of media and state when each would be used, explain the features and uses of the complex, defined, selective, and differential media, and provide examples of how to culture anaerobic microbes. All right, so let's get right into it. And so here we have a picture of several different types of media, um, and it kind of demonstrates that we can actually grow microbes in many different ways. Now, the type of media that we use is going to depend on what we actually are trying to do. And so we can classify media both by its physical state, so is it solid, liquid, or semi-solid, its chemical composition, and its function. So let's kind of break that down a little bit. And so we have semi-solid media. This is often used in motility testing. You can see it in many different ways where you can actually have semi-solid tubes where microbes can grow through the media. So it has some agar in it, which will help them grow. We've got broth media, which we've seen a little bit in class, like when we did our bologna exercise. This is going to have nutrients. It's going to be in a flask or a tube. And in pretty much all cases, our media is going to be sterilized. This is useful when we just need to grow a bunch of an organism. It can also be used for enriching for specific types of organisms, but the goal here is usually just growing a bunch of a thing. We have solid media, which, solid and semi-solid, they're very similar. They're going to have agar in them. The amount of agar is going to determine how solid they are. And it's going to be sterilized, just like everything else. And then it's going to be poured into a plate, or it's going to be poured into a tube. So when we put them in plates, this is usually going to be used for isolating individual colonies or being able to view individual colonies. Semi-solid media can oftentimes be used for pour plates, where you want to not have it quite as thick as you would in a regular agar plate. Now we can put these things, whether it's liquid, solid, or semi-solid, into tubes. Here we have we have two different types of tubes. The Simmons citrate is a regular solid agar, and then this motility test is a semi-solid agar. They can be done in slant format like this, where there's a slanted area. It gives us more surface area to streak an organism on, or a deep, and these are used for stabbing into the medium. And so again, while the slants, I'm not familiar with semi-solid slants. I'm only really familiar with solid slants. When it comes to semi-solid media, this could be both when it comes to deeps where you do the stabbing, it can be solid or semi-solid. And again, here, I'm not trying to get you to memorize every aspect of the different types of media and like memorize this. I just want you to kind of be aware of how the media are used. So slants, we're going to streak that area. We have more surface area to do our streaking. In deeps, we're going to stab because we want to figure something out like motility or something like that. Now, we can also talk about chemical composition. Now, most of the media that we use in labs for like micro classes are going to be complex media. These are going to be things like LB or tryptic soy or nutrient agar or nutrient broth. And so these are going to have like some very complex things that are in there, such as tryptone or peptone or yeast extract. And then they might have some other things that are defined. Now, defined media are going to have a very specific concentration of all organic and inorganic compounds. So we're going to know exactly what's in there. We know, for example, in this glucose minimal salts media that it's got 20% glucose. It's got uh, this specific amount of magnesium sulfate, calcium chloride, so forth and so on. And so we use defined media oftentimes when we're trying to grow specific types of organisms, or if we're trying to see if organisms can grow on specific carbon sources and stuff like that. um, and so this is just showing you that our defined media are chemically defined, so we know exactly what's in them, every organic and inorganic component. And as I was saying, like, it can be used for some heterotrophic organisms. It's often useful for autotrophs. A lot of the nitrogen, like when you're dealing with organisms that are nitrifiers, so they convert ammonia to nitrate, they might be grown in a medium like this. I've used it in the past when I am trying to grow an organism on a specific carbon source. So if I want to know what organism can grow on in the presence of a plastic or something, that's the only carbon source there, then I have a defined media, and then I add in the plastic as the carbon source. On the other hand, our complex media are going to grow most things that can grow on plates and in broths. This is going to be a mixture of a bunch of stuff. They're not going to be fully defined. You're going to have these ingredients like tryptone and peptone, which are broken down, proteins or partially decomposed proteins. And these are oftentimes going to have very, broad arrays of what can grow on them, including some of your fastidious organisms that have complex growth requirements. So you have all these different things available to them so they can grow. Now, you may also hear this called enriched media as well. Now, one of the things that we use in both the clinical lab and in research lab are differential and selective media. So these can be used for selecting or differentiating between different organisms that are in a sample. So this might be something like a fecal sample or a urine. And this can help clinicians determine what organism is causing an infection. And so we'll start with differential media. This is a way to differentiate between organisms that have the ability to perform some function. And so here we have blood agar. Blood agar is a differential media. It doesn't prevent anything from growing, but you can distinguish between the organisms based on how they grow. So here we have 3 different organisms. We have one that is doing alpha hemolysis, one that's beta hemolysis, and one that's gamma hemolysis. So what we're doing here is classifying organisms based on their ability to break down blood. The alpha hemolysis partially breaks it down, beta hemolysis completely breaks it down, and gamma hemolysis doesn't break it down at all. And again, the important part here isn't for you to memorize the differences between these 3 types of hemolysis. The important part is to understand that differential media allow you to distinguish between different organisms based on their function. Now, we also have selective media. This allow us to select for specific types of organisms. So they often contain something in the media that will actually suppress growth of other organisms. We have 2 examples here, MSA and EMB. And so I'm just going to show you kind of how these can be used. So in mannitol salt agar, it has high salt content. It's about 7.5% NaCl. And this is going to differentiate. So it's also differential. It differentiates between organisms that can ferment mannitol, but it only selects for organisms that are able to grow on 7.5% salt. So what we see here is, for example, both of these are able to grow, right? So it didn't select against them. So we know they're able to grow on 7.5% salt. But the one that changed the media to yellow was able to ferment mannitol. Now, we also have eosin methylene blue, which is also a selective and differential media. And this one, it has these two dyes, eosin and methylene blue, which will intercalate into the gram-positive cell wall, and it prevents them from growing. And it also differentiates on their ability to ferment lactose. So we can assume that all three of these are probably gram negatives because they grew quite well. We've got a streak here, here, and here. But only these two were able to ferment lactose. We can tell that based on the color change. Again, I'm not really worried about you being able to differentiate between lactose fermenters and non-fermenters, just kind of understanding what we're looking at and how to interpret it. And so we'll practice this in class a little bit to see how those questions will look. Lastly, most of the stuff we've been talking about is just, I kind of think of as what we do when we're growing aerobic organisms. But there are anaerobic organisms out there that are not going to grow in the presence of oxygen, right? And so we talked a little bit of that with oxygen growth requirements. And on average, like in certain types of wounds, you might see up to 38% of the population of that wound being anaerobic. And when you're trying to grow these things, you have to be very careful because exposure to oxygen will kill them. And if you remember from the other lecture, it's because they don't possess enzymes to deal with those reactive oxygen species. And so we have special types of tubes that are used. So like when you're taking samples as a practicing nurse, you might need to collect a wound sample in a specific tube to transport it to the lab so they can culture for anaerobes. Once they're being cultured, there are a couple of different ways to do this. You can add thioglycolate to the media. You might use something like this anaerobic jar. Regardless, the key is that to grow anaerobes, you need to remove oxygen. Another way you can do it is in the presence of or inside of an anaerobic growth chamber. This is going to be a big chamber that has like glove holes that you can put your hands in, and it prevents oxygen from entering it. You displace all the oxygen with CO2 or nitrogen, and it's going to look something like this. And so on the right-hand side over here, we've got these this little chamber, this entry chamber. So this is where you go in, and then you close this door, and then you open it from the inside using this glove box to actually move the sample around outside of from environmental into anaerobic area for you to do all your work. All right. So with this, you should be able to describe the three formats of media and when they would be used. So this is like your physical states. You should be able to explain the features of and uses of complex, defined, selective, and differential media, and provide examples of how to culture anaerobic microbes. All right, I hope you enjoyed, and I will see you all soon.